I don't always listen to podcasts, but when I do, I listen to STEM Talk, interviewing the most interesting people in the world of science and technology. Stay curious, my friends.
Welcome to STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society. I'm STEM Talk host Don Carnegas, and also joining us is Dr. Ken Ford, IHMC's director.
Hi, Don. Great to be here with you on STEM Talk.
So today we have a super interview that Ken and Dominic D'Agostino recently conducted with Dr. Marc Matson. I played hooky on this one, but left it in their able hands. Dr. Matson is a leader in understanding the cellular and molecular mechanisms underlying neuronal plasticity and neurodegenerative disorders, and has made major contributions to understanding the pathogenesis of Alzheimer's disease, Parkinson's disease, ALS, and stroke. In addition to his interest in discovering the underlying mechanisms of these diseases, Mark is focused on prevention and treatment. In this episode, Mark, Ken, and Dom get into lots of fascinating topics to include longevity, intermittent fasting, ketones, ketogenic diets, protein in the diet, Alzheimer's, Parkinson's, sarcopenia, and much else.
Intermittent fasting has become very popular these days, and Mark Mattson is, in our view, the premier authority on this matter.
But before we get to today's interview, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we especially appreciate all the wonderful 5-star reviews that are piling up on iTunes. Keep them coming.
As we announced in several earlier episodes, the Double Secret Selection Committee has been carefully and continually reviewing the iTunes reviews with an eye toward selecting the best reviews to read on STEM Talk. There are extra points available for pithiness, wit, and of course, lavish praise, our favorite.
And if you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk shirt. Today, our winning 5-star review was posted by someone named Carl. As soon as we hear from Carl, a STEM Talk shirt will be on the way to him.
And here is Carl's winning review. Really smart, really interesting people being interviewed by the same. IHMC is a fascinating place and attracts like-minded people. The guest list is as eclectic as the subject matter. It's no accident that people operating at the very top of their professions happen to be really interesting.
Thank you, Carl, for the great review, and thanks to the thousands of other STEM Talk listeners who have helped make STEM Talk a success right out of the gate. Okay, now on to today's interview with Dr. Marc Matson.
Dr. Marc Matson is Chief of the Laboratory of Neurosciences at the National Institute on Aging. He is also a professor of neuroscience at Johns Hopkins University. Mark has published more than 700 peer-reviewed articles in leading journals and books, and has edited 10 books in the area of neuronal signal transduction, neurodegenerative disorders, and mechanisms of aging. Dr. Matson is among a handful of the very most highly cited neuroscientists in the world. Dr. Matson has done seminal work and produced major advances in the fields of neuroscience and neurodegenerative disorders.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Welcome to STEM Talk, Mark. Good to see you.
It's nice to be here, Ken.
Our pleasure.
Dr. Matson, when did your interest in aging begin?
It really began in 9th grade when I was writing an essay for a class I had, and I decided to write it on cryopreservation, cryonics. And, this is 9th grade. And
Very interesting.
that's still from the standpoint of human science fiction, but the idea is that you can preserve the body and brain forever or indefinitely and then bring back to life, so you could stop aging that way. But then from a scientific standpoint, I really got interested in aging when I was doing my postdoctoral research, which was in developmental neurobiology. And we were studying the mechanisms that regulate the formation of neuronal circuits during development. And also we were studying cell death, because it turns out that during brain development, many more neurons are initially generated that end up surviving. And so as the circuits are refined, neurons die. So that got me interested in cell death. And on the other end of life, old age, neurons can die, particularly dramatic fashion in Alzheimer's, Parkinson's disease, and so on.
Well, how then did you go from there to your present position at the NIA?
Okay, so my postdoc, after getting my PhD at the University of Iowa, I did postdoc research in Colorado, and there we were discovering that neurotransmitters, which at that time, this is in the late 70s, early 80s, were thought to act only at synapses in the adult brain. We found they were playing an important role in guiding the growth of axons and dendrites and in the formation of synapses. And so then I discovered, for example, that the main excitatory neurotransmitter in the main brain, which is called glutamate, can regulate dendrite outgrowth. I got interested in how proteins called neurotrophic factors can interact with neurotransmitters to sculpt, if you will, the formation of neural circuits. And then we got interested in cell death because it turns out if glutamate receptors are overactivated, can actually kill neurons, and that's very important in conditions such as stroke, for example, when cells don't have enough energy, or in traumatic brain injury. Then if they continue to be excited when they don't have enough energy, they may die. So then I was recruited to the University of Kentucky, and I was on the faculty there in Lexington for 11 years, and that's where I really got my own research program going in the Sanders-Brown Center on Aging. Colonel Sanders and John Y. Brown, who's a former governor of the state.
Amazing.
Yeah.
So much of your research is focused on the benefits of fasting or intermittent energy restriction, especially on reducing the risk of neurodegenerative diseases. So, and you touched on it a little bit. What are some of the mechanisms by which this works?
Yeah, so the simple notion is that if you challenge yourself and your cells, including your brain cells, intermittently, bioenergetically, either for exercise or fasting, that the nerve cells respond adaptively and pathways are activated that increase the resistance of the neurons to more severe stress and perhaps resistance to age-related neurodegenerative disorders. So this all started when I was at Kentucky, and since we were interested in aging in general, it had been known for a long time that one way to increase the lifespan of rats and mice is to reduce their energy intake. And whereas most labs do that by reducing daily calorie intake, counting calories each day, we started subjecting animals to alternate day fasting, where one day they have no food for 24 hours, the next day they can eat again. And if you repeat that for beginning when the animals are young, they'll live up to 30% longer. And then we found we have many different models that are relevant to neurodegenerative disorders like Parkinson's, Alzheimer's, and even acute degenerative conditions like stroke or severe epileptic seizures. And we found that rats or mice maintained on this alternate day fasting diet, their nerve cells were more resistant to degenerating, and the functional outcome was better in the animals on fasting compared to animals that ate every day.
And just to clarify, the days that they're eating, it was ad libitum.
That's right.
Right.
And their overall calorie intake, though, is reduced somewhat. In other words, they don't eat twice as much food on the days they do have food as they normally would. So they're about anywhere from 10 to 25% calorie restricted.
Does that vary between strains of animals, too?
It does.
Okay.
Yeah.
That's interesting. I understand that you conducted a National Institute on Aging study that looked at two groups of obese women at elevated risk of breast cancer in the UK, where the women eating a 200-calorie-a-day diet twice a week fared better than the women eating a calorie-restricted daily diet. Do I have that right? And could this 5-2 approach be useful for people in other conditions?
This study was done as a collaboration between Dr. Michelle Harvey at the University of Manchester and my own lab. And she's a dietitian that has a strong interest in breast cancer, and so she saw our work in animals, and we talked about a human study. And so what was done is that about 100 women were divided into 2 diet groups. And after getting information on their basal diets and their estimated calorie intake, we divided them into 2 groups. One group, they ate 25% fewer calories each day by counting calories at each meal. The other group, we had them 2 days a week, and in this study was consecutive days, eat only 500 calories. And this was a 6-month study, and more than 80% of the women made it through the 6 months, and most of them had dropped out because of the diet, dropped out in the first month. If they got by the first month, they were losing weight, feeling better, and they stuck with it. The take-home message from the study was that the women on what's now called the 5-2 diet, they lost more belly fat, and they retained more lean mass, and they also had bigger improvement in glucose regulation as indicated by increased insulin sensitivity compared to the women who were counting calories each day. And that's consistent with what we know about fasting in terms of general energy metabolism. If you eat 3 meals a day, plus snacks, as a lot of people do in this country, every time you eat, the energy goes into your liver, glucose goes into your liver, it's stored in the form of glycogen, and that's always tapped into first. And it takes upwards of 10 hours to completely deplete glycogen stores. So most people never deplete their glycogen stores, but what happens then if you go longer, say 12, 16 hours or more, not eating anything, then you mobilize fatty acids, and they go into the blood, into the liver, and converted to what are called ketones. And ketones are a really good energy source, alternative energy source for cells, including neurons. So we think that these differences we saw were mainly due to this energetic shift where the women on the 5-2 diet were, if you will, burning fat, whereas the women who just reduced daily calorie intake but ate regular meals didn't.
Following up on that, could everyone, not just people with identified health risks, therefore benefit or potentially benefit from fasting?
There haven't been controlled trials in humans done in people who are not overweight, to my knowledge, and so this is needed. What I can tell you, though, is that based on animal studies, we think it's very likely that even animals and possibly humans who are normal weight may show some benefits from intermittent fasting, particularly for the brain. And from an evolutionary perspective, it makes sense that if you're hungry and haven't been able to figure out how to find food for a while, that your brain better be functioning well. And we find this is clearly true in animals, and we have some insight into actually what's happening in the nerve cell circuits and at the molecular level that explains why the animals on intermittent fasting diet have better cognitive performance, even if they're not old and don't have a disease.
Please discuss, if you would, various approaches to intermittent fasting. You know, one sees compressed feeding windows or every other day fasting. There must be some pros and cons, and also do you see a role in intermittent fasting for exogenous ketones or for medium-chain triglycerides? Could you talk about this topic sort of generally, how you recommend an intermittent fast be structured?
Yeah, well, I don't make any specific recommendations, but I can tell you that there are, as far as human studies, there's been 3 different approaches. The 5-2 diet, alternate day fasting, which is actually alternate days, eating only about 500 or 600 calories, and that's by Chris DeVerdi at University of Chicago, Illinois, Chicago. And then the other approach is what's called time-restricted feeding, where you limit the time window each day that you take in calories to about 6 to 8 hours. There's an investigator at the Salk Institute, Sachin Panda, who's a circadian rhythm biologist, and he did some nice work in animals, first showing that if you restrict the time period that they have access to food to 8 hours every 24 hours, you can feed them a high-fat diet, and they won't get fat compared to the animals that have access to food 24 hours. Interestingly, even though both groups eat the same amount of food. And then he recently did a study where he had a postdoc who wrote a cell phone app, and then they gave this cell phone app to like 150 free-living subjects out there, San Diego area, and they were instructed to, every time they ate something or drank something, take a picture of it. And then the picture went to the postdoc at the Salk Institute, and then it didn't stay on their phone. And what they found, they had a paper in the journal Cell Metabolism recently where they found like 80 to 90% of the subjects put something caloric in their mouth from the time they got up until they went to bed. In other words, they get up, eat something, eat lunch, snack, eat dinner, eat something in the evening. Pretty much the only time they weren't taking anything caloric was when they're sleeping. So they're going like 14, 16 hours they're eating something. So the only time they're fasting is when they're sleeping. So we think that, and this is occurring obviously on the background of obesity in this country, is occurring with people with these kinds of eating patterns. So we had a workshop on this 2 years ago, and then we published a paper in PNAS based on the workshop that talks about, kind of summarizes the research having to do with meal frequency and timing and health and what's known, what isn't known, what are myths. For example, that breakfast is the most important meal of the day and that you're not going to be healthy if you don't eat breakfast. There's other kinds of, if you will, myths. For example, that it's healthier to eat a lot of small meals rather than eat one or two big meals. And largely this isn't based on any good science that we can find. And so if listeners are interested, they can track down that article.
Great. Do you see a role for ketogenic fats or exogenous ketones when coupled wisely, carefully with a intermittent fasting?
Possibly. Fasting can elevate ketones to levels that are pretty high and actually higher than you get with a typical ketogenic diet. Now, some of the more recent approaches that are being tested mainly experimentally have to do with ketone esters and diesters. And these will elevate ketones up into the range you get with fasting. But as far as adding on ketogenic diets or, for example, ketone ester supplementation with fasting, studies need to be done. I can't really say until the studies are done. And this is kind of generally true with this whole field, that there have been quite a few animal studies done, a few controlled human studies done, a lot of anecdotal things floating around on the internet, but much more needs to be done in rigorously controlled trials.
And sort of on that topic, you discussed the benefits of ketones in your TED Talk. And to what extent do ketones, particularly beta-hydroxybutyrate, account for some of the beneficial effects of fasting? Can you speculate on that?
That's something we're very interested in, particularly from the brain aspect. And it's been known for a long time, and in my TED Talk I put up a slide in Roman times and Greek times when people had epileptic seizures and were having convulsions. They thought they were possessed by demons, and they found if they shut them in a room for a few days, the demons go away. And very likely what's happening is they're fasting, the hydroxybutyrate, acetoacetate levels are going up, and that's suppressing the seizures. So we know that, and as you know, ketogenic diets are still used in some patients with epilepsy, and they work. So it's known that hydroxybutyrate and acetoacetate can be metabolized to acetyl coenzyme A and then used to produce ATP in cells, including neurons. So that's kind of the conventional thinking of how these ketone bodies are an energy source. But recent findings from other labs and our lab as well suggest that, for example, beta-hydroxybutyrate has signaling functions. Other labs, Eric Burden, for example, has shown that hydroxybutyrate can inhibit certain protein deacetylases. We have found recently, and presented this at the Society for Neuroscience, and we're revising a paper, that hydroxybutyrate can increase production of a neurotrophic factor, a protein that promotes the growth and survival of neurons. It's called BDNF, brain-derived neurotrophic factor. And we and others had previously shown that both exercise and fasting increase BDNF levels in the brain. In fact, BDNF, we think the scientific community, neuroscience, thinks is a key mediator of the antidepressant effects of exercise, as well as the most commonly used antidepressant drugs, the serotonin reuptake inhibitors. So we find that hydroxybutyrate increases BDNF production, and it's known that BDNF is important for learning and memory. In animals, if you block BDNF signaling, it will cause cognitive impairment. BDNF is important for a process called neurogenesis, the production of new neurons from stem cells in certain regions of the brain. So the point there is that I think that, and the way I look at this, again, going back to an evolutionary perspective, okay, if you get this shift in energy metabolism where you've depleted the glycogen stores, and the animal or us are saying, okay, we better figure out how to find food pretty soon, that your brain better be working well. And in addition to providing an energy source, these ketones may be a peripheral signal that goes into the brain and stimulates signaling pathways that improve cognitive function.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research lab pioneering groundbreaking technologies aimed at leveraging and extending human cognition, perception, locomotion, and resilience.
Following up on this, a 2012 issue in the journal Neurobiology of Aging, a team of researchers led by Kashiwaya, and including Richard Veach at the NIH and yourself, published a very interesting paper reporting that ketone esters had cognitive sparing properties and lessened amyloid and tau pathologies in a mouse model of Alzheimer's disease. Do you see a role for exogenous ketones in enhancing brain health or performance?
Yes, this is an important area of research from the standpoint of Alzheimer's disease and age-related cognitive impairment. There's interest in doing controlled clinical trials to test the efficacy of. In fact, there's one going on now with coconut oil. There's one going on with the branched-chain amino acids. We are interested in doing a small study with the ketone ester in that animal study you mentioned, which we were part of. It was actually done in my laboratory by a postdoc that worked with Dr. Veach. And so I think this is an approach that really needs to be tested sooner rather than later. As you probably know, that Alzheimer's disease, there's been no treatment that is effective in slowing down the disease process. There's a few drugs that have a very modest transient beneficial effect in improving cognition, and they're widely prescribed, but they don't have a big effect, and they don't affect the neurodegenerative process. With these age-related diseases, the biggest impact will be on risk reduction, identifying what people can do during their midlife that will reduce their risk from developing these disorders when they get in their 70s or 80s. So I'm not so optimistic that we're going to cure, if you will, Alzheimer's disease when someone's symptomatic, because there's already tremendous degeneration of nerve cells. On the other hand, I'm very optimistic that through diet, lifestyle, perhaps some sort of dietary supplement and pharmacological, science-based pharmacological interventions with safe approaches that, and identifying people at risk is another thing. We're all at risk in a sense because the data now are if you live to be into your 80s, you almost have a 50% chance of having Alzheimer's disease before you die.
Yeah, that's a depressing statistic. Have you thoughts on other neurodegenerative disorders like Parkinson's with respect to fasting or ketone esters?
Yeah. So there's good evidence that particularly exercise, a lot of studies in human Parkinson's patients that exercise programs they benefit from. We've done some work on intermittent fasting in Parkinson's models in mice, and the intermittent fasting has beneficial effects. The issue that comes up with clinical trials in patients who are already symptomatic, I think is a valid one that once they're diagnosed, they usually start to lose body weight, both in Alzheimer's and Parkinson's disease. In fact, in many cases, they're starting to lose body weight before they're diagnosed. So again, I think that focusing on risk reduction is the approach to take. But from a mechanism standpoint, ketones will benefit the neuronal populations that are vulnerable and degenerate in Parkinson's disease, just as they benefit the neurons involved in learning and memory that are more vulnerable in Alzheimer's.
Sarcopenia, the loss of muscle mass, strength, and function, is just a major problem associated with aging. Only last month, a new article was published in Nature that seemed to show that autophagy is a critical regulator of stem cell fate, with implications for fostering muscle regeneration in sarcopenia. Autophagy typically declines with age, and this may cause stem cells to lose their so-called stemminess and become senescent. Both intermittent fasting and ketosis increase autophagy. Might this account for some of the common benefit that one sees in both of these approaches?
Yes. The way we look at this is that during the bioenergetic challenge, whether it's vigorous exercise or fasting, and I should point out that the ketones would, with extended endurance exercise, ketones go up too, that those bioenergetic challenges will increase autophagy in part by inhibiting a pathway involving a protein called mTOR. And what's happening is the cell is going into kind of a protective mode. It's reducing overall protein synthesis and at the same time enhancing its ability to remove molecular garbage. So, for example, proteins that have been damaged by free radicals or mitochondria that aren't functioning well. So during the bioenergetic challenge and possibly in response to the ketones, the cell is enhancing its ability to cope with stress. But then, very important for the cells to grow, whether it's muscle cells or nerve cells, is a recovery period. So after fasting, eating. After exercising, resting and eating. So what happens then, the cell, having gone through the bioenergetic challenge of exercise or fasting, is cleared out the garbage, and it's ready to grow, produce new proteins and grow. And so during the recovery phase, then there's increased protein synthesis and growth of the muscle cells. In the case of neurons, growth of dendrites and even formation of new synapses, which we and others have documented occur with intermittent fasting diets and also exercise.
That's a great answer. Thank you. With an eye toward longevity and avoiding sarcopenia, what strategies can be employed, and I think you've touched on this, to optimize muscle strength, while on an intermittent fasting protocol. And do you think it's possible to actually gain muscle while intermittent fasting?
Yeah, it is possible to gain muscle while intermittent fasting. Intermittent fasting is not necessarily reducing calories a lot. So for example, if you do the time-restricted feeding where you eat only during an 8-hour time window each day, one approach is that you, for example, skip breakfast. This is kind of an easy approach because then you've already fasted for the time you've been sleeping. And then exercise, maybe late morning, and then eat after that, and then eat a normal amount of food, during your 8-hour time window. Have a healthy diet, which is important. So you're not going to lose muscle mass as long as you maintain your energy intake. From the standpoint of aging, one aspect of diet that I think is important and should be discussed a lot more is protein intake. There's quite a bit of evidence that high protein intake is not good for aging, maybe not even for your muscles. And I mentioned this mTOR pathway. So if you take in a lot of amino acids, that will increase mTOR and it will reduce autophagy. It will inhibit the ability of cells to clear out the molecular garbage. It's a little bit counterintuitive because, the conventional wisdom is if you want to build muscle, you got to take a lot of protein in. And that may be advantageous in the short term, but for lifelong maintenance of muscle mass, my own opinion, although further work needs to be done, is that it's probably not a good idea. And in fact, if you look at a lot of these bodybuilders, you could make the argument, maybe they're taking testosterone or something, but many of them, when they get older, they start to have a lot of health issues cropping up.
One sees branched-chain amino acids and essential amino acids often advocated to an aging population as a part of the means of avoiding sarcopenia. And so your recent comment just now is sort of in opposition to that.
Not so much. I think what you want to do is get the ketones up, but not necessarily have really high levels of proteins.
Right. And by proteins, you're including the amino acids in general.
Yeah.
Right.
Yeah.
Of course.
Do you fast? And so I'd like to hear a little bit about your regimen.
Well, what I do is a time-restricted feeding type approach. 4 or 5 days a week, I don't eat breakfast or lunch. Then I'll work out mid-afternoon and then eat right after that, and then eat some more in the early evening. And then the weekends, I'm home with it. And this is just what fits into my work day and lifestyle. And I think I'm more productive this way because I'm fortunate, I guess. I don't have to go to breakfast with people or have to go to lunch with people most days. And so I'm able to just get into the lab early in the morning. And I drink a lot of green tea. So I've found that it's not a problem for me. I don't get hungry in the morning. Start getting a little hungry before I exercise and then eat right after I exercise. And I've maintained the same, I've done this for years, and I've maintained the same body weight for actually 30 years.
Wow.
Within a few pounds.
And you go into your exercise fasted, in a fasted state, no supplement.
That's interesting. I do the same thing. I have an early dinner, skip breakfast, and go into resistance training, usually fasted.
Yeah.
You might want to say something about what you perceive as the benefit of fasted training.
Yeah, well, pretend you're a muscle cell. Okay, so you got to think like a muscle. Okay, you you've been experiencing lower end levels of glucose for a long time, 16, 18 hours. Amino acid levels are kind of low because you haven't eaten anything for 16, 18 hours. Then you're asked to work. Then you work. So what would be your best response to ensure that next time this dummy whose body you're in finds food, that you're able to take up the nutrients and utilize them to your best advantage? And the answer is you upregulate glucose transporters in your membranes. You upregulate amino acid transporters so that you can quickly move the amino acids in when the person is done tracking down the prey and eating it. So that's the idea. There's an evolutionary basis of why this makes sense, that you should be functioning well both cognitively and physically in the fasted state, because that is the conditions that, for example, carnivores use to survive. If your brain wasn't working well when you're hungry, and if you weren't physically fit enough and have endurance enough to track down your prey, you're not going to survive. So, the 3 meals a day plus snacks is very abnormal from an evolutionary perspective, and there's no really solid scientific basis of why eating 3 meals a day might be the healthiest. In fact, we don't think it's the healthiest approach.
Do you when you do eat, do you try to choose foods that will maintain and preserve that nutritional ketosis, or do you just try to get the benefits from fasting alone?
That's a good question. The answer is no, I don't. Maybe I should think about that more. But I eat what I think is a healthy diet: a lot of fruits and vegetables, nuts, meat as fish, yogurt. But I do eat whole grains, oatmeal and whole wheat, and probably I'd say, and beans. I'd say at least half my calories are from complex carbohydrates, either whole grains or beans.
Do you avoid meat at all?
I don't eat. I don't meat. I tend to avoid red meat.
Red meat. Okay. Exercise is a powerful stimulator of brain-derived neurotrophic factor, BDNF. How does exercise elevate our moods? I think you kind of touched on that, but yeah.
Yeah. Well, so when you exercise, we think there's changes that are occurring directly in the circuits of your brain. Many nerve cell circuits are more active, obviously the ones involved in sensory motor function. But also, it turns out those involved in cognition the nerve cells themselves are more active during exercise. And it's been known for a long time that activity in neurons increases productions of neurotrophic factors, including BDNF. And, for example, I do a lot of trail running. And during trail running, you have to be focused on every footstep you take, or you're going to hit a root or rock and fall down. And so, again, going to evolutionary perspective, the antidepressant effect of exercise seems to involve some of the same mechanisms that are responsible for the cognition-promoting effects of exercise, including increased activity in the neural circuits. As you know, one of the old treatments for depression was electroconvulsive shock. Okay, so that's a little extreme form of increasing activity in neural circuits. But during exercise, there's a much milder increase in activity. But on the other hand, some of the same mechanisms are going on involving BDNF. And as I mentioned, the most commonly used antidepressants are thought to have their antidepressant effects through BDNF. BDNF increases neurogenesis, increases the number of new neurons, and increases the number of synapses, particularly in the hippocampus, which is a brain region that's affected in depression.
Interesting. So there's an activity-dependent increase in BDF, but also you alluded to the potential for a ketone-induced. Okay, very interesting.
I don't always listen to podcasts, but when I do, I listen to STEM Talk, interviewing the most interesting people in the world of science and technology. Stay curious, my friends.
You know, in the past, you've mentioned historical figures with, really good brains, high-performing individuals who promoted fasting. Could you tell us a little bit about that?
Yeah. Well, the first thing I'll tell you is I found this book called The Fasting Cure. And it was written by Upton Sinclair and published in 1911. And many of your listeners may know of Upton Sinclair because he wrote a book called The Jungle, which exposed some of the problems in the meatpacking industry around the turn of the around 1900. And so in this book, The Fasting Cure, Upton Sinclair interviewed about 200 and some people who had some ailment. They went on fasts of various lengths and intermittent nature, and almost all of them had improvement in whatever ailment they had. But then if you go back as far as cognition specifically, Plato in his writings you can find where he wrote that he did some of his best thinking when he was fasting. Certainly people like Gandhi. And as you know, in many religions, fasting is a common theme. And part of it, I think, is because you're able to think more clearly in the fasted state and perhaps contemplate things you might not have contemplated when you're in a more satisfied, from a energetic standpoint state. And then there's a lot of quotes you can find. For example, Mark Twain, he commented that something to the effect that, that the physicians make 3/4 of their money on essentially people who overeat, and that fasting, therefore, like 75% of physicians would lose their jobs if people were to fast regularly.
Totally agree with you. So if fasting is so good for us, why have we been told to eat 3 meals a day? And even as of late, why are we told to eat kind of mini meals throughout the day? So why is this information being given to us?
Well, I think that the 3 meals a day probably started, I'm not an expert on this, but in what I've read, started with the agricultural revolution where people were working very hard farming every day, and it was a practical approach. They had a high energy demand because they have a lot of physical labor, so they eat when they got up, then they'd work hard for 4 or 5 hours, take a break, eat more, discuss what work they're going to do in the afternoon, work more, then eat some more. And then that just kind of stuck with it in civilized societies, agricultural societies. Then as far as eating a lot of small meals, my impression, at least one impetus for that advice came from work with diabetes patients and with the knowledge of insulin and the role of insulin in glucose regulation. And the idea there is that the thinking was it's important to avoid big spikes in glucose. So if you just eat a lot of small meals, then you'll get, more frequent, smaller increases in elevation in glucose, and maybe that's better for you in the long run. But on the other hand, it's very clear from experimental studies that many small meals is not as effective as intermittent fasting in improving insulin sensitivity. And then as far as breakfast, one aspect of that has to do with studies done in school-age children where they will take kids who most all normally eat breakfast, and then one day they'll take half of them and have them not eat breakfast. The other half will eat breakfast. And then midday they do some behavioral tests, and they find, lo and behold, the kids who didn't eat breakfast are more irritable and can't concentrate on their work as well. Well, yeah, they're hungry, but I would submit if they had never eaten breakfast,
adapted to that.
If they were
regimen,
adapted, if they were adapted to it, they'd be fine and would do as well or better than the other kids.
So how long does it take the average person to adjust to a regular fasting regimen, and could this be a treatment for obesity?
Yeah, in the studies we did with Michelle Harvey, and also we did a study in asthma patients within a month. It seemed like 3 weeks to a month seems like a tipping point. The women in the study in England that led to the 5-2 diet, and we gave them questionnaires. They could score their mood and their hunger, rate their hunger, and so it was about 3 weeks to a month. By then, their mood was improving, and they were no longer hungry on the days they were having very low energy intake. So it's kind of similar to exercise. If you've been sedentary and then try to go run 5 miles, you're not going to feel very good. It takes a while for you to get in shape. I would propose that it also takes you a while to get in shape for fasting.
The more you do it, I think, the more. So out of curiosity, what was the longest time you ever fasted?
Just one day.
Just one day?
Yeah.
I'm curious if you take any supplements too, if you think supplementation is important.
I don't anymore.
Okay.
I was on board with the idea that antioxidants are important and free radical scavengers, but actually 25 years ago it became pretty clear that that's not right, and in fact we published a paper in Scientific American this last year where we point out the emerging evidence that many of the chemicals that in fruits and vegetables that are good for our health may be good for our health not because they're free radical scavengers, but because from an evolutionary perspective they're toxins. The reason they're produced by the plants is to dissuade us and insects from eating them. So they typically have a bitter taste, and this is true for many of the actually supplements you get now, which I think actually that may be good. So there's certain supplements like sulforaphane is one from broccoli, or curcumin, or even caffeine, the most commonly consumed toxin. Caffeine's a toxin. You can kill yourself with caffeine, and it has a very bitter taste. And so we think that during evolution, our cells and organ systems adapted so that we could cope with the potentially toxic effects of these chemicals and still get the calories and other nutrients from the plants. And so if people are interested, they can read that July 2015 issue of Scientific American for that. And
some do appear to derive some benefit. You mentioned drinking green tea. And there's been a ton of research on EGCG, for example.
Yeah, the epicatechins.
So,
and dark chocolate.
And dark chocolate, yeah.
But again, if you take, everyone knows this, you take dark chocolate, 100% cocoa, it's very bitter tasting. Most people can't eat 100% dark chocolate. And the epicatechins in tea, if you buy the powder epicatechins and put it on your tongue,
it's awful.
It's very awful.
Yeah, make a gag.
So again, these are chemicals that have a mild noxious effect. And interestingly, that during evolution, we developed enzymes in our liver that rapidly metabolize all these naturally occurring chemicals. So they're only in our system a short time period, and then they're removed. So we think they induce a mild adaptive stress response, then they're removed. Kind of a transient stress response similar to transient exercise or possibly even fasting. Yeah.
Could you talk a little bit more about mTOR? As you noted, there's lots of evidence that reducing mTOR is beneficial with respect to longevity, yet mTOR seems critical for muscle protein synthesis. And as we age, as we discussed earlier, sarcopenia is part of the puzzle. And are we doomed to sort of a balancing program here, or would it be that you intermittently depress mTOR or intermittently elevate mTOR? Could you just talk a little bit about that?
Yeah, I think your last point is right on that. The ideal scenario seems to be intermittently inhibiting mTOR so that you can intermittently increase autophagy and other mechanisms for removing the damaged molecules. But it's very important to also intermittently activate mTOR so that the cell can produce the new proteins that it needs to grow and adapt and function better. And so I could envision in the future that, in contrast to the sort of current thinking from the pharmaceutical industry and drug development, where you identify a target and then you develop a drug that you can get to stay like at a constant level in the blood, and so you're constantly affecting whatever pathway. So I think from the standpoint of optimizing health, that pharmacological approach would be to develop interventions that you would administer intermittently, either to inhibit mTOR intermittently, or perhaps there's other things. We've worked a little bit with 2-deoxyglucose, which sort of tricks cells into experiencing the state of caloric restriction or fasting, if you will. And we find in our studies that cells are most healthy, if you will, when they're intermittently exposed to 2-deoxyglucose versus more continuously exposed even to a relatively low level of 2-deoxyglucose.
Exercise certainly seems to work
and
that
exercise,
way.
That's right.
That's a great answer, Mark. Could you tell us a little about your role at the National Institute on Aging?
Yeah. So my title is Chief of the Laboratory of Neurosciences. So I'm in charge of a small group of scientists of about, we probably have 40 in our group, but only 3 of them are senior scientists. And so the organization of research at NIH is such that each institute, National Cancer Institute, National Institute on Aging, et cetera, are divided into what are called laboratories. So a laboratory at NIH is equivalent to a department at a university. And a laboratory at NIH is headed by a chief, which is equivalent to a chairman or chairperson in a department at a university. So the organization is similar. The terminology is a little different, so people unfamiliar with it, say lab chief. Well, lab chief is kind of like a department chair, although the department's smaller than a department at a university.
That's great. And is there anything else that you'd like to talk about where we're together here?
No, I guess not.
We covered it.
Yeah, we covered a lot.
I have one
Okay.
more question. What are your thoughts on short-term fasting? For example, you said this intermittent kind of 2DG, and it made me think of someone who would do a 3-day or 5-day fast maybe once every month or 2.
Yeah.
I know people that are doing this for cancer. Maybe
Yep.
it would push your body to purge precancer cells. So what are your thoughts on that?
I think it's a good idea worth pursuing. One of my colleagues, Walter Longo at USC, who we wrote a review article on fasting together last year, that he's using that approach, and he's most interested in cancer. This 5-2 diet, Michelle Harvey now is doing a study where women who have breast cancer, when they're receiving chemotherapy and radiation therapy, they will be placed on either the 5-2 diet or a control diet during the entire course of their therapy. And the idea there is that most cancer cells rely on glucose as an energy source and are not good at using ketones. So, if you're fasting, if you're fasting and the cancer cells are being bombarded with toxic chemicals or radiation, they're much less likely to survive. And therefore, we think you'll be able to have a bigger impact on eliminating the cancer. And that's what's been shown in animal studies with intermittent fasting. It will, by itself, slow greatly the growth of tumors, and in combination with chemotherapy or radiation therapy, can wipe out the cancer cells much better than those treatments alone. Also,
maybe protect the normal cells.
Exactly.
Too.
That's
Yeah.
yeah. The normal cells, since they can use ketones, they are responsive to the fasting in terms of adaptive stress responses. They may be more resistant to the adverse effects of the cancer treatment, so maybe less side effects.
Well, thank you very much. It was a great interview, and I appreciate you doing it.
Yeah.
So thank you again.
Yeah, well, thanks for your great questions, Ken.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Wow, that interview that you and Dom did with Mark Mattson was absolutely full of great information, clear, concise, and in-depth.
Mark continues to be an impressive thought leader in literally every area to which he turns his attention. An impressive fellow. Dom and I could have talked with him for hours, but we restrained ourselves as his travel was delayed and he didn't arrive at IHMC until the wee hours in the morning. And right after this interview, he had a major public lecture to give at IHMC. Mark is a trooper and clearly a resilient guy.
That must be the consequence of his intermittent fasting.
Absolutely.
I invite you to visit the STEM Talk webpage where you can find the show notes for each episode. We have provided links to 3 of Mark's most relevant papers and to his recent public lecture at IHMC. You can find us at stemtalk.us. This is Dawn Carnegie signing off for now.
And this is Ken Ford saying goodbye until next time.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others. So please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.